Hello, Dr. Manila. Here is my eReport for the cellular biology portion of this course. The first topic I'll be discussing is the cell membrane. The cell membrane is the cell's barrier to the chaotic world around it. It allows for organized biochemistry, and without it, life wouldn't be possible. It is not only the barrier, but is also the gatekeeper of the cell. The lipids in the membrane act as the barrier, while the proteins act as a portal into the cell. The cell membrane is also known as the phospholipid bilayer. It is unique because the membrane is made of amphipathic molecules. The head groups are hydrophilic, while the hydrocarbon tails are hydrophobic. The structure of these phospholipids causes the hydrophobic tails to huddle together because they're trying to hide from the aqueous environment outside of the cell as well as inside of the cell. This forms two opposing layers known as the bilayer because both sides of the membrane are hydrophilic, while the inside of it remains hydrophobic. One phospholipid is a head group and a tail group, but the two tails oppose each other, which is how it forms this phospholipid bilayer. These hydrophobic properties allow the membrane to be able to spontaneously form and self-seal without requiring energy. The opposing layers creates asymmetry between the two faces, though this cannot be seen in the diagram. The outer face, which is what faces the extracellular fluid, has much larger phospholipid heads than the inner face, which faces towards the cytoplasm. This actually promotes the circularization of the membrane. The cell is able to control and regulate the fluidity of its membrane. If the hydrocarbon tails lack double bonds, the phospholipids can come closer together and pack closer together, lowering the fluidity of the membrane. But if double bonded carbons are in the hydrocarbon tail, it will result in kinks in the tails, making more space between each phospholipid. This will increase membrane fluidity. Even if there are kinks in the hydrocarbon tail, membrane fluidity can actually still be regulated and altered. This is because the gaps that are formed with the kinks in the hydrocarbon tail can actually be filled with cholesterol. This is denoted in the diagram by showing these yellow structures that are sitting in between the phospholipids. When these gaps are filled with cholesterol, it actually makes the membrane denser and much less fluid. Due to the delicacy of the cell membrane, it is crucial that it be properly supported. From the cell's interior, the cell membrane is lined with a cell cortex. This actually strengthens the cell membrane. The membrane also contains many different types of protein. If a protein is embedded into the membrane, as shown here, here, and here, it's considered to be an integral protein, while other proteins are considered peripheral proteins that are simply bound to the outside of the membrane. An example of a peripheral protein can be shown here. This peripheral protein is held to the membrane by binding itself to an integral protein. There are also transmembrane proteins, which are defined as proteins that extend fully through the membrane with hydrophilic regions on both heads. They must be hydrophilic on the extracellular side and the intracellular side, because if not, it will reject the aqueous environment and revert to the center of the membrane, and it wouldn't be transmembrane. The membrane is a great barrier, but things must be able to move in and out of the cell. Small nonpolar molecules are able to easily diffuse through the membrane from a high concentration to a low concentration without the use of a protein. Uncharged polar molecules can diffuse if they're small, but the larger they get, the slower diffusion gets. Also, all ions and charged molecules are blocked from diffusion across the membrane. They require help to get through the membrane. Transportation across the membrane is controlled by transporters and channels. Channels, shown here in the diagram, are able to achieve specificity through charge and size. Only the right size and properly charged molecules are able to pass through the channel. Channels do not require any energy to function. They just sit there in the membrane and act as a portal for those molecules to diffuse through, so they can enter or exit the membrane. Now, transport proteins that are found in the membrane transport a single molecule and are extremely specific. An example of a transporter protein is shown here in the diagram. To be able to pass through a transporter protein, the correct molecule must bind, causing a shape change in the protein that allows it to cross the membrane. Transporters also may require energy to be able to function. They can be responsible for passive transport, also known as facilitated diffusion. This is when a molecule that's following its concentration gradient is moved from a high to low concentration. And its concentration gradient is what's driving its transport. Transporter proteins are also responsible for active transport. This involves forcing a molecule to move against its concentration gradient through the use of ATP. Only specialized transporters called pumps are able to accomplish this. An example of this would be the movement of sodium. The interior of the cell actually has a lower concentration of sodium than the outside of the cell. But if this transporter protein was a sodium pump, a sodium ion in a low concentration would bind to the transporter protein. This binding causes a shape change in the sodium pump, and this change in shape will actually force the sodium ion out of the cell into a higher concentration. This is how sodium is able to be moved against its concentration gradient. Vesicles are able to bud and fuse to organelles in the endomembrane system. This includes the cell membrane, ER, the Golgi, lysosomes, and endosomes. Their compatible membranes allows for this. Vesicles are the way the cell transports molecules and proteins, whether it be in a secretory or endocytic pathway. The formation, docking, and fusion of a vesicle is a complex process that requires many different proteins. Most vesicles are covered in a protein coat. For the purpose of this report, the focus will be on clathrin-coated vesicles. Each vesicle begins with the formation of a clathrin-coated pit, which is this first image in the diagram. Clathrin, which is the protein denoted in the dark green, is a protein that forms the protein cage around the vesicles and helps maintain its shape. Another class of the protein coat proteins are the adaptins, which is denoted in light green. Adaptins ensure that only the right cargo gets into the vesicles. The adaptins interact with the clathrin to secure the clathrin to the vesicle membrane. There are also cargo receptors that are vesicle membrane-bound proteins. These are denoted in blue. Each type of adaptin can interact and bind to specific cargo receptors, and this is how vesicle contents are sorted. The proteins that travel in the vesicles have specific transport signals on them, and it is these signals that bind to the cargo receptors. Once everything is properly bound, the cargo is bound to the cargo receptor is bound to the adaptin, and the adaptin is bound to clathrin. Eventually, a small protein called dynamin, which is this protein in yellow, forms a lasso around the neck of the vesicle, pinching off the vesicle from the membrane and releasing it. After the vesicle has been released, motor proteins walk along the cytoskeleton, carrying the vesicle to its proper destination. After the vesicle has been walked to its location, it now must be docked to the target membrane. There are Rab proteins on the surface of the vesicle. Rab is what tells the cell what membrane that vesicle originated from. There are also tethering proteins on the target membrane surface. The tethering protein and Rab are able to interact non-covalently. It is this interaction that secures the vesicle to the membrane without fusing. This is known as docking. After docking, the vesicle must fuse. And for the vesicle to fuse, it must become extremely close to the target membrane. There is a V-snare protein on the vesicle, and there's a T-snare protein on the target membrane. V for vesicle, T for target. When these two interact after docking, V-snare and T-snare then twist tightly together, bringing the vesicle closer and closer until the two touch and fuse. Once fusion occurs, the cargo of the vesicle can either be released into a different organelle of the cell or to the extracellular environment. It all depends on if it uses the secretory or endocytic pathway. The fusion of a vesicle to its target membrane can actually be delayed and will be stopped after docking to await for the proper cellular signal. It'll just wait there at the target membrane, ready to fuse and release its cargo, but won't do so until it's told to. This is very common in neurotransmitters. The cytoskeleton of the cell is dynamic. It has the ability to change and move. It allows for cellular division, differentiation, organization and positioning of organelles, crawling, and muscle contractions. It even acts as the cell's transportation system. The cytoskeleton is made up of 3 types of specialized protein filaments: intermediate filaments, microtubules, and actin filaments. The intermediate filaments, as shown here, provide tensile strength in order to protect the cell against any type of mechanical stress, like pulling, squeezing, and/or stretching. The intermediate filaments are able to make fragile things more durable. These are the toughest of all 3 of the filaments in the cytoskeleton. Most of them are anchored to the nucleus and then extend into the cytoplasm and outwards to the cell membrane. Intermediate filaments are formed by individual fibers that come together to twist into strands. The strands then come together and twist once again into a rope-like fashion to form an intermediate filament. The intermediate filaments can then dimerize, and those dimers can then come together to form tetrameres. The protein that makes up intermediate filaments contains heads that are globular. These globular heads are able to interact with other intermediate filaments, anchoring proteins, and organelles. The second class of cytoskeleton proteins are microtubules. Microtubules are long, dynamic, hollow tubes that are able to rapidly assemble, disassemble, and then reassemble. Microtubules are more functional rather than structural when compared to intermediate filaments. This is because they are used as the highway of the cell, allowing for transport around the cell. All of the microtubules originate at the centrosome and extend out throughout the cell. Microtubules are made out of tubulin dimers. These dimers are made up of 2 globular proteins, alpha and beta. These dimers stack to form protofilaments that alternate between alpha and beta subunits. It actually takes 13 protofilaments lining up next to each other to make one hollow microtubule. Microtubules also have polarity. These ends are denoted as plus and minus. Both ends of the microtubules can have dimers added to it, but it is more effective when adding to the plus end. This is why, shown here in the image, only the assembly and disassembly are happening towards the plus end. The centrosomes contain ring structures that act as a nucleation site, as seen here. The minus end of the microtubule gets embedded into the centrosome, while the plus end extends outwards into the cell periphery, where dimers can be added to grow the microtubule. Microtubules have dynamic instability, which means they are constantly disassembling and retracting back to the centrosome. They can disassemble partially or completely. Dimers are more effectively added to the growing tubule when bound to GTP. Because they are added faster, they are more stable and are able to hold the microtubule together. If the polymerization of the microtubules is too slow, the GTPs are cleaved into GDPs, lowering the dimer's affinity with the other dimers in the tubule. The GTP-bound dimers are denoted in darker colors, while the GDP-bound dimers are denoted in a lighter color. If the dimers are added quickly, the dimers that are bound to the GTP act as a cap, holding the microtubule together, preventing it from fraying. This is known as the GTP cap. The cap can be seen starting here, all the way extending out to the growing end of the microtubule. As it grows, the GTPs are constantly being cleaved in the microtubule, but as long as the GTP cap is there and is still extending outwards with the microtubule, it'll remain stable. When the plus end of the MT is able to anchor to something else in the cell, it'll stabilize it. If not, the GTP cap will get smaller and the MT will end up falling apart. As shown in part C of the diagram, there's no more of those darker units left. They're all those light colors because they're only bound to GDP, and their affinity is so low, the microtubule falls apart. Actin filaments are the final class and are essential for cell movement. For example, crawling on surfaces, cell division, and mitosis. Actin filaments themselves are unstable until other proteins bind to them, which stabilizes them. Depending on what proteins bind, actin filaments can be permanent and stiff, or temporary and dynamic. The actin filaments are thin and flexible, and they're made out of a twisted chain of actin glomerular subunits that are all identical. All of these subunits are pointing in the same direction, therefore actin filaments have polarity. There's a plus end and a minus end. A growing actin filament can be extended on either end, but it is faster and easier to add on to the plus end. If the actin monomers are bound to ATP, it allows them to be added to the growing filament more easily. Once added, the ATP is cleaved, and this reduces the strength of binding. This is actually a good thing because actin filaments must be dynamic. This allows for easy assembly and disassembly. Typically, half of the total actin proteins in the cell are assembled into filaments, and the other half are still monomers. They remain monomers because other proteins are bound to them, but once the cell needs more actin filaments, the proteins simply let go to promote assembly. These actin filaments are extremely versatile. They can even be used to alter viscosity in the cell, or they can be used to help provide mechanical strength to the cell membrane and increase its durability. But they're also crucial in cell crawling. Cells depend on actin to be able to crawl along surfaces. They are able to sense extracellular cues and migrate towards it. Crawling is able to take place in 3 steps, and actin takes a critical part in each step. Step 1. A large extension known as lamellipodia is pushed out of the leading edge of the cell. This is a dense sheet formed by a meshwork of actin filaments, forcing the membrane outwards. This can be seen here in the upper right portion of the diagram. The actin is positioned where the plus end is facing towards the leading edge of the plasma membrane, allowing for actin polymerization. There are also smaller projections called filopodia. These are made up of parallel bundles of actin. These can be seen here in the diagram. These actin filaments are parallel to one another, pushing out. These are filopodia, but lamellipodia is this meshwork of actin. Both are made up of actin, but are just organized differently. Lamellipodia and filopodia are constantly being sent out by a crawling cell. Only when they sense a chemoattractant will they begin to anchor to the surface. Step 2. Now the extensions must bind to the surface they're trying to crawl on, but they must be able to bind in a way that is reversible. There is a transmembrane protein called integrin that adheres to molecules in the extracellular environment. Integrin is a bridging protein because they're intracellularly anchored to actin and to the extracellular surface being crawled upon. If in a multicellular organism, the cell will crawl along the extracellular matrix. Integrins will link to a protein called fibronectin, which is bound to collagen. This is how they're able to anchor themselves to the extracellular matrix. The third and final step. The cell is now anchored, but it must pull the rest of itself along the surface. Actin is a part of the entire inner coating of the membrane, also known as the cell cortex. Myosin, which is a type of motor protein, is also bound to actin, and using contractile forces, it's able to pull the cell cortex, which then pulls the entire cell forward. A shape change then occurs in integrin, making it let go of the surface, allowing these steps to occur again and again until the cell has reached its target destination. The G-protein coupled receptors, also called GPCRs, are a 7-pass transmembrane surface protein that carries signals into the cell through a series of protein modifications. GPCRs play a role in almost every aspect of cellular communication. When a signaling molecule binds to the GPCR, it causes it to change its shape. This shape change in the receptor will actually activate the G protein that is associated with the receptor. There's a wide variety of G proteins, but the one thing they all have in common is that they can all bind to GTP and GDP. When the GPCR and the signaling molecule bind, the complex will transmit that extracellular signal to the interior of the cell. This is why it is known as the primary messenger. When the signal is then passed from one intracellular signaling molecule to another, those are known as second messengers. The GPCRs are what trigger that signaling cascade. The G protein itself is made up of 3 subunits: alpha, beta, and gamma. When the protein is inactive, the alpha subunit is bound to GDP. But when the receptor is activated, the alpha subunit lets go of GDP and binds to GTP. This then causes the alpha subunit and the beta-gamma subunit to separate from each other. Once they disassociate, they are now able to function independently of each other. The G protein is able to activate enzymes and open ion channels that are found within the membrane. Some specific enzymes that they are able to activate are adenylate cyclase and phospholipase C. The G protein itself is actually capable of shutting itself off. The G protein has GTPase activity, which allows it to hydrolyze GTP into GDP, inactivating itself. When the G protein is activated, the alpha subunit is able to bind to the enzyme adenylate cyclase that is found within the membrane. This activates adenylate cyclase, which, once activated, is capable of converting ATP into cyclic AMP. This enzyme faces the interior of the cell, and cyclic AMP is able to act as the second messenger. It is the presence of cyclic AMP in the cell that conveys the original message from outside of the cell. The cyclic AMP is then able to activate an enzyme within the cell known as protein kinase A, or pKa. pKa is then able to phosphorylate target proteins around the cell. It can even enter into the nucleus and phosphorylate transcription factors. This phosphorylation is what is going to change the cell's behavior in response to that original extracellular signal. The G protein can also activate the membrane-bound enzyme known as phospholipase C. Phospholipase C is then able to hydrolyze a membrane component called PIP2, causing it to split into IP3 and DAG. DAG stays in the membrane and activates protein kinase C, or PKC. PKC can then phosphorylate target proteins, one of which is calcium channels. This opens the channel, allowing calcium to flow into the cell from the extracellular environment. The other component of PIP2, IP3, does not remain in the membrane. It actually travels to the interior of the cell. IP3 will then enter into the smooth endoplasmic reticulum and trigger it to release calcium into the cytoplasm. Calcium, the second messenger, can now bind to PKC, increasing its activity. It can also bind and activate the protein calmodulin. Calmodulin can then wrap around CAM kinase, activating that enzyme, which is now phosphorylating other target proteins. This phosphorylation is what is going to change the cell's behavior in response to that original extracellular signal. Cell division is a complex process that is closely regulated. This is also known as the cell cycle. The cells are able to cycle through the cell cycle, which consists of 4 stages: G1, S phase, G2, and M phase. There are proteins required for all of the major events in each phase, and they must be turned on and off in the proper pattern in order to achieve the correct cellular effect and at the right time. This cyclic regulation is typically controlled by phosphorylation and dephosphorylation. Phosphorylation and dephosphorylation is the cell's on and off switch, and this allows the cell to be able to turn on and off all proteins and enzymes needed for each phase of the cell cycle. Instead of remaking all of these proteins and enzymes for each stage of the cell cycle, the cell keeps the proteins present, but inactivates them. When they are needed, all the cell has to do is activate them, and not remake them. Typically, kinases and phosphatases accomplish this. The question becomes, how do the right proteins get activated and deactivated at the right time during the cell cycle? The actual activity of these enzymes are staggered, which is possible through cyclins. Cyclins are a small set of signaling proteins that control kinases. The kinases they control are known as cyclin-dependent kinases, or CDKs. When a particular cyclin is made, its corresponding CDK becomes active. The CDKs themselves are not made cyclically, but the cyclins are. The S-cyclin is made right before the S phase begins in order to activate the S-CDKs. The S-CDKs not only activate the other proteins necessary for S phase, but they also phosphorylate the G1 phosphatases, turning them on. The G1 phosphatases will then go on and turn off all of the G1 proteins. As the cell is transitioning from G1 into S phase, S-cyclin is not only starting S phase, but it's also turning off G1 phase, allowing for a smooth transition between the phases. There's also an expression of M-cyclin that begins at the end of S phase and increases during G2. During this time, it's binding to its corresponding CDK. These are just some examples of how the cell is able to achieve the major events in each phase of the cell cycle. CDKs are also able to arrest the cell temporarily. The G1 checkpoint checks for induced mutations in the genome. If there is DNA damage, there will be an increase in the transcription factor p53. p53 then interacts with the mediator complex to turn on the gene for p21. p21 then binds to G1 kinase, stalling the cell cycle in G1, preventing it from entering S phase. This buys the cell time to perform nucleotide and base excision repairs. This is critical because if the DNA is not repaired, it'll be replicated during S phase, and that mutated DNA will be passed on to daughter cells. This is why it's so critical to halt the cell cycle, repair the DNA, and then move on to S phase for replication. Also, if the DNA cannot be repaired, p53 causes the cell to undergo apoptosis. The cell would rather die than pass on mutated DNA to its daughter cells. Once successfully through these checkpoints, the cell is now prepared to divide. First, the centrosome must be copied, and the two must move to opposing poles of the cell. This duplication is triggered by S-CDK and G1S-CDK during the beginning of S phase. Also, M-CDK phosphorylates and inactivates microtubule binding proteins, increasing the dynamic instability of the microtubules. This allows for the centrosome to send out more microtubules more rapidly during mitosis. The cell cycle is a delicate and intricate process. But through the cycling expression of cyclins and the activation and deactivation of CDKs, it allows for all of the right proteins to be on and off when necessary for the cell cycle to be able to progress smoothly. Epithelial cells, when bound together, almost act like our own cell membrane. Epithelial cells make up the skin, intestinal lining, and so many other structures. They act as a barrier while allowing specific import and export of things. The epithelial sheets have polarity to them. Each side is different biochemically and structurally. The outwards-facing side is the apical surface, and the inward-facing side is the basal surface. The basal surface of the epithelial cell is connected to the organism by attaching to the basal lamina. Epithelial cells connect to other things through cell junctions. There are 3 overarching categories of cell junctions, and there are 5 types of cell junctions total. Tight junctions are where 2 cell membranes are so close to each other that nothing is able to pass through them. Even small, water-soluble molecules are unable to pass through. To form a tight junction, there are proteins called claudins and occludins on either side of the cell junction, shown on this side as well as on this side. These proteins act like two sides of a zipper, and when it is zipped up, it seals the two cells together. These tight junctions also define membrane domains because membrane proteins cannot laterally diffuse through claudins or occludins. Tight junctions are critical for life to occur because without them, life would not be possible. For example, it is critical that hydrochloric acid be produced in the stomach for digestion, but it also does not reach any other part of the body because if it did, it would cause catastrophic damage. And a tight junction is what makes this possible. 3 out of the 5 cell junctions fall under this category of mechanical junctions. These cell junctions are adherens junctions, desmosomes, and hemidesmosomes. The overarching category of mechanical junctions are classified by their function, which is holding cells to other things. They also provide mechanical strength by joining the neighboring cells' cytoskeletons together. Adherin junctions and desmosomes bind epithelial cells together, and hemidesmosomes bind epithelial cells to the basal lamina. Adherin junctions and desmosomes are able to accomplish this through the use of a transmembrane protein called cadherin, which interacts with the cytoskeleton components. In adherin junctions, the cadherin interacts with actin filaments, as shown in this upper portion of the diagram. And in desmosomes, the cadherin interacts with keratin, which is a specific type of intermediate filament. Now, hemidesmosomes use a different transmembrane protein than adherin junctions and desmosomes. It uses the transmembrane protein integrin to interact with collagen in the extracellular matrix, to bind and stabilize the epithelial cells to the basal lamina. The final type of cell junctions are gap junctions. Gap junctions are openings formed between two cells, but only if they have matching transmembrane protein complexes called connexons. When the connexons match, they come together to form a channel between the two cells. This channel now permits for small inorganic hydrophilic molecules to diffuse between the two cells. Gap junctions are also regulated and can be shut or opened in response to cues. This could be caused by a change in membrane potential or ligand binding. A living organism is highly coordinated and regulated because every cell should be working for the greater good. Normal cells are community-oriented and are very social. Normal cells care for the greater good of the multicellular organism and will do whatever necessary to accomplish that. For example, this could be dividing when more cells are needed, or halting division when there are enough cells already present. Cells should always maintain their biochemical and cellular characteristics, and stay where they belong in the multicellular organism. Cells will undergo apoptosis when the cell has lived for as long as it has needed to, or it has become defective. A normal cell will always sacrifice themselves for the greater good of the organism. Each cell lives and thrives to participate and contribute to that multicellular society. The cells are told what to do through signaling molecules, gene regulation, and phosphorylation. Now, cancer is antisocial and will violate all of these rules. Cancer cells are rapidly dividing, selfish. They will invade and destroy healthy tissues, and they do not care about the organism. If one cell breaks any of these rules, it's not that big of a deal. But if that cell enters into the cell cycle, there's a major problem. This can result in a mass of asocial cells that has no compassion or empathy for the greater good of the organism. Cancer cells are defined by 2 main properties. They must be rapidly proliferative. No developed human cells are able to divide as fast as cancer cells. And they must invade or colonize space that belongs to other cells. This diagram shows how cancer cells will invade space, not follow the proper cellular structure, and will expand past the boundaries of the tissues. Also, the cells sticking out are at risk of becoming metastatic. This means that the tumor has migrated to the blood or lymph systems, which then carries it to other parts of the body. Normal cells are supposed to stay within their tissue of origin, but if they do happen to travel, they actually cannot survive in different tissues. But when cancer metastasizes, wherever it lands, it'll be able to give rise to secondary tumors, because they can survive in any tissue, even if it's not their tissue of origin. Due to cancer's rapid division, it allows for rapid evolution. This gives them the opportunity to outcompete the healthy cells, and that alone can allow them to survive better than the healthy cells. This rapid evolution can result in supercells, which makes them able to degrade the extracellular matrix, attract blood supplies, and give them the ability to enter into blood or lymph. These are just some of the properties, but there are many other properties that make cancer so dangerous. Cancer cells no longer need any signals from other cells to survive and function. Also, cancer cells may not default to apoptosis. This is because they are asocial and do not care about the survival of the organism, just their own survival. They will never sacrifice themselves just for the greater good of the organism. And with this, they can even be immortal. Cancer cells will do whatever they need to do to survive. They will never consider the greater good of the multicellular organism due to their antisocial behavior.